Hello, everyone, and welcome to this audio podcast. This audio podcast as part of a Quagmire interlude. This audio podcast as part of a Labonix interlude. This audio podcast as part of a Bason scroll, and this audio podcast as part of a Storm Robotics production. In this audio podcast, interlude, into this audio podcast, there is a in this audio podcast interview a little bit in regard to looking at the effects of experimental psychology and physiological stress on the immune function. So in one investigation, psychology was induced. Psychological strain was induced by confronting human volunteers with semi-realistic and ambiguous and potentially uncontrollable interpersonal scenarios. 50 male recipients aged between 24 and 55 years were exposed to a theoretically irrepressible sort of stress scenario. Participants were required to solve a challenging problem, and consequently they were requested to identify a solution to a confederate to a confederate to a confederate to the investigators. The investigational teams investigational sort of teams prompted sort of a mild sort of strain as recognized by mood variations in the investigational cohort in the investigational cohort when required with a control group of 36 male participants. Blood was taken through a catheter before directly and after, and 15 minutes after and 13 minutes after the strain or after the challenge. Numbers of cell types were established, and in this investigation, the team summarized that strain short term strain and stressors short term strain and stressors are restricted to cells of the natural killer cell population, NK cell population. Stressor control and irregularity had demonstrated to be critical in the psychological and physiological physiological outcomes. Stress triggers the hypothalamic-pituitary-adrenal axis, or HPA axis, raises flowing glucocorticoids, and it's concomitant with variations of immune cells and predisposition to infections. Opioid peptides, and catecholamines are part of the reaction to challenging situations. It had also been distinguished that stress, for example, is linked to the suppression of immune cells, and there is a confirmation that immune cells may not adapt or cope over time, which is consistent with Hans Selye's 1936 with Hans Selye's 1936 general 1936 sort of general adaptation syndrome model, and in particular in around point three of the general adaptation syndrome model. Both sort of taming and taxing situations prompt biological effects that can alter the development of the immune modulated pathophysiological movements a movement, modulated pathophysiological movements, and the association between psychological characteristics and immune responses to an acute stressor had also been investigated. Sudden strain sudden strain and physical activity has demonstrated to augment immune reactions. Psychological stress, such as trauma, shows an intense circulation in concentrations of epinephrine, norepinephrine, beta endorphin, adrenocorticotropic hormone or ACTH, cortisol, and selective distribution of NK or natural killer cells into blood. Similarly, an investigation looked at the association between sudden strain and extensive sort of physical activity to identify the influence and solidity of adhesion molecules and other cell types. In the investigation, participants were settled and a catheter was implanted into the forearm. They were then asked to relax for up to 30 minutes. In order, participants then completed a 15-minute verbal communication challenge, and then a 15 to 18 minute physical activity challenge where the investigators utilized cycling as one of the challenges, and there was a 60-minute duration between end of each challenge. Whilst seated, participants are organized 3-minute dialogue on 3-minute dialogue on paper, and then presented on an imaginary sort of scenario. The full time of a verbal challenge included 6 minutes of planning and 6 minutes of performance was 6 minutes of performance, and was about 15 was about a total of around 15 minutes. For the cycling challenge, participants were told that it would be up to 15 to 18 minutes, and starting with a sequence of 3-minute durations marked by cumulative resistance, and it would be a larger effort. The investigators had found that both strain and physical activity has an important relationship on moving T cells. In another investigation, where a team looked at acute strain and long-term restriction strain in animal models, and particularly the Sprague-Dawley rats, on T cell subsets to identify whether glucocorticoids can establish modification in cell reallocation. The outcomes from the specific investigation informed that number one, stress changes white cell circulation. Number two, that adhesion molecules could be convoluted in stress-induced modifications of T cell scattering, and three, that variations could be linked to flowing glucocorticoids, and also to sort of coping and adaptation with frequent challenge or challenges. That draws this audio podcast to a close. This audio podcast is part of a Quagmire interlude. This audio podcast is part of a Labonix interlude. This audio podcast as part of a Bason scroll. This audio podcast as part of a Storm Labonix production.